responsibly. Okay, so other questions? Alrighty then. So what we're going to do here is we're going to talk quickly about diabetes, and then we're going to get into signaling, and the signaling is related to kind of what we're going to talk about here for diabetes. So in terms of diabetes, what we've got is diabetes is it's the disease where your body does not sense blood sugar. Okay, and we've got type 1 diabetes. Type 1 diabetes, it's also called early onset diabetes. Essentially, the body is not making insulin. So early onset, this happens and you see this in kids or young adults. And if it's untreated, it's lethal pretty quickly. Okay, so the example, I'm not going to really talk about it, but they had, when they did the first test of this in humans, once they figured out it was insulin, first they did it in a dog, then I think it was just one dog, but they gave a dog diabetes, essentially, by removing its pancreas, and then they gave it insulin to save it, okay? And then they had a 14-year-old boy or something who had diabetes, essentially was going to die in a couple weeks. They gave him insulin and completely recovered. Okay, so that was like the first test where they did that. Type 2 diabetes. This is the body becomes resistant to insulin. So there's insulin around, but what's happening is cells don't recognize it. The liver doesn't see the insulin. Okay, and the mechanisms for that are kind of complex, but essentially you've just got receptors that don't recognize it. So something that can happen in diabetes, I've got pictures down below, is you can end up with neuropathy, meaning you end up with deadened nerves. Okay, that's one of the dangers of insulin. So in other words, you can't feel your feet. And so you get these awful pictures. This is not nearly as bad as some of these pictures of people. But essentially what you've got is somebody could step on, that's a big, right? That's their picture for emphasis. Somebody's walking along, you could step on pins and you wouldn't know it. Okay, but more than that, what you actually get is more often than that, you'll get something that's called a diabetic sore, what you see on the foot here. And what's going on there is when we're standing, we're going to stand here, we're going to get uncomfortable on one foot, you can stand on another foot. But if you can't feel your feet, you'll stand on the same spot all the time. Eventually that causes you to get a sore. So that's what's going on there. I actually have neuropathy in my feet and ankles from my fall, but mine is different in that, so neuropathy means the nerves aren't working correctly, right? I just have constant pain, so I can feel things. I can actually feel things through that, but it's weird in that I have to look a lot to make sure that what I'm feeling is it real or is it not, essentially. But yeah, we've got a diabetic sore here, and here we have somebody whose foot was crammed into something or other. Maybe it was their shoes got too small or something, and over time they couldn't tell because they couldn't feel their toes. Okay, so that's one of the things that happens. Now, I'm not going to go into it too much, but it is actually related to our next topic here. So what you end up with is you end up with problems in the blood vessels. So one of the things that happens is we have this signal molecule called nitrogen monoxide. Okay, so you have changes in like blood cell, like capillary thickness and things. And so there's various things that happen here that you end up with less nitrogen monoxide being produced. Okay. So nitrogen monoxide is, and so that can lead to what we're seeing here, the neuropathy. The neuropathy is it's not a big blood vessel problem, which you end up with in diabetes as well. It's a capillary problem in that you're not getting enough oxygen to those nerves, and essentially they die and they stop signaling, they die, I think. Yeah.
You said one of the dangers of insulin. Did you mean one of the dangers like without insulin?
Oh, one of the dangers, right? One of the dangers of diabetes. Really. Thank you. Okay, so just in diabetes, you're ending up with dysfunction in your capillaries because of that, because of the glucose. It's caused by having high glucose. So what I want to do is then talk about, so what NO is going to signal. So you end up with deficient NO signaling. We'll talk about what it signals in a second here, but NO is going to signal that capillaries should dilate, right? So if you have capillaries, they're down there and they're constricted all the time, then your nerves aren't going to get all the nutrients they need. That's where you get that from. Okay, so this NO was the molecule of the year in science in 1992. Which stands out to me because that was like one of my first years of grad school. I remember reading about it and things like that. But yeah, this was the cover of Science that year. And what goes on with NO is a great molecule. People are like, how is that a neurotransmitter, right? It's just an NO. And you're going to actually get it off of arginine. Get it off of arginine in terms of cells doing it. But NO, if we look at it and think about it as, go ahead and do a Lewis dot structure of it like that. There's NO. What is it that might help it go through cell membranes? Yep, super small. Just 2 atoms, right? Okay. And what else? If we go over to the periodic table, you guys have memorized all your electronegativities, right?
Totally, for sure.
This is about 3. This is about 3.5, right? We call something a non-polar. What's the difference? What's our line for non-polar? It's 0.4. It's actually like the official textbook definition. But that's just somebody drew a line, right?
Yeah.
Somebody drew a line and then they put it in the textbooks, and so all as professors say, okay, there's a line. But it's not a line, it's a continuum. So 0.5 isn't very big, right? So it's pretty non-polar. Okay, so it's pretty nonpolar, so it goes straight through membranes. All right, so it's a great signal molecule. It is a short-range signal molecule. You don't get this made in your blood and it goes everywhere. So we wouldn't call this a hormone. I'm going to see what I have down here. Okay, there we go. So I'm going to have to I'm drawing structures. So let's talk about, so if we have NO, what it's going to do is it takes so you have NO, it's going to interact with an enzyme that makes that causes cyclic GMP to be made. Okay. So we could think this enzyme would be a guanylyl. You don't need to memorize this. Cyclase. Okay. So you get a guanylyl cyclase, you make cyclic AMP, and that cyclic AMP, or cyclic GMP, I'm sorry, the cyclic GMP is going to cause the dilation of blood vessels. Okay, does that make sense? So this is the cyclic, we're talking about signaling now. Cyclic GMP is a second messenger. So it's a molecule inside the cell that gets made that causes a various set of things to happen. All right. So it's not, so often, not in this case, often you have something bind to the outside of the cell, like let's say insulin, and that causes something inside the cell to be made that signals to everything else. And we'll talk about what the second messenger is for insulin in a bit, but not yet. Make sense what happens there?
So what's the second messenger for ML?
For a number of things could cause, so when cyclic GMP gets made, depending on the cell type, the cells will do things. In the case of blood vessels, we're going to cause them to dilate. Okay. I don't know what all the other things that other tissues do, but it's like cyclic AMP, same sort of thing. Something will signal it to be made on the outside of the cell. It gets made, and then different cell types are going to do different things based on having made it. Okay.
Is that word about cycles?
Guanylyl. Guanylyl. But you don't need to know that so much. You need to know that NO crosses the membranes easily, and it will go into the cells and cause things to be active, cause cyclic GMP to be made. But it is interesting talking about this. So there's a number of things in medicine that result. So we talked about angina before. That's heart pain, right? You have pain in your chest because the heart isn't getting enough oxygen. And angina will be treated with nitroglycerin. Trying to decide if I was trying to remember if I told this story first semester. So the way they discovered this is that it wasn't that. So Ben that worked in dynamite factories, ammunition factories, where you're working with nitroglycerin around. When they went to work during the week, they had less chest pain, and when they went to on the weekend, they had more chest pain. Okay, so what happened is they were getting exposed to some amount of, after seeing enough people like this, some physicians figured out, okay, maybe there's something to do with that, and it turns out that nitroglycerin can break down to make NO, which causes their blood vessels to dilate, which causes more blood and oxygen to be able to get to the heart. So if we look at nitroglycerin, this is our structure. It's essentially glycerol with nitro groups on it. And so if you have chest pain, you can have some pills and you will take these. This is a really quick effect you get. You take the pills and you can get a relief from that. You can get your blood vessels dilated pretty quickly. So this is going to break down to make NO, and that's going to go on to cause blood cell dilation. I always want to spell.
Blood pressure.
What was that?
Is it also blood pressure medication?
It might be. Mostly I've got, so it is reducing your blood pressure when you do the dilation. It's just not going to be a long, I mean, I think you'd have to be constantly taking the pills, as I understand it. Does anybody know anybody that takes this? I don't know anybody who's taken this. My understanding is it's a, right now, acute, take it to take care of the pain sort of thing. But if you were going to be using it, you'd have to be using it. You have to take it and then take it. You might take it pretty frequently. It wouldn't be one of these once a day things.
Yeah.
That's my best. I'm not sure. Okay. Questions? All right. Well, then the other fun story with this that apparently got slipped down below are base things we were learning about. Well, way down at the bottom. Let's talk about this one. So let's talk about Viagra. And I think I've actually mentioned this one to you before.
Yeah. They had a very fun conversation about whether it would work in.
See, I don't even remember that very well, but I've taught this too many times. All right, so what happens with, so Viagra, we talked about this then. What it does, though, is I think it's interesting to go over it again because we're talking about signaling, right? So we have NO, right? And that's going to cause cyclic GMP to be made. So actually, let's do it like this. Let's not even use G. Let's not even worry about NO. So we've got GTP, right? And that can be made into cyclic GMP with a guanylyl cyclase. All right, but if your blood vessels would stay dilated all the time if this didn't go away, right? So something has to tear this down so that the blood vessels will respond. They'll dilate and they'll constrict. And so what happens then is that can be broken down to make GMP, sorry. And that's done with a phosphodiesterase.
Is this just like an example or do you want to say no?
This is an example. Okay, but it's an example of how you can make drugs to do things. So Viagra, and I pull this out of my old notes so I don't get the picture of the woman with the football sitting on the bed all the time. What am I getting? I'm in YouTube, I'm getting the, whatever these blue chew things are now all the time. Have you seen this? Okay. I was like, what is this? Oh, okay. But anyway, so what Viagra does is it inhibits the breakdown of cyclic GMP. Okay, so how might that cause blood vessel dilation?
Everything stays cyclic GMP.
Yeah, everything keeps cyclic. You got cyclic GMP; it doesn't break down, and so the blood vessels stay dilated. This while Viagra's in effect. And then we talked about this how really what this was intended to be was a treatment of a blood pressure treatment, right? Because you got like 600,000 Americans a year die from heart disease, so big market there. And it turned out that it only actually dilated blood vessels in the penis, and they figured out that, well, they could make money doing that with that too. But you could imagine we could either activate this or we could inhibit this. And most of the drugs I know about are actually inhibitors, right? You know where the active site is. You stick something in the active site, you screw it up, right? Or you bind something to it. I don't know many drugs that activate things. Questions? Okay. Doesn't mean it can't happen.
What chapter is this?
This is chapter 10, I think. Other people agree with that?
Yeah, well, that's the end of chapter 9.
That was the end of chapter 9?
19.
Okay.
Diabetes was with 19.
Diabetes is 19,
This
but the NO is more related to chapter 10. Okay, so chapter 10 here. So these pictures right here are going to be actually, they're from a different book. They're from Leninger. So you won't find these pictures there, but I like them better than what we've got in your book. So what I want to talk about here is we've got 4 characteristics of signaling systems. And so wherever we see receptor here, we're most likely, not always, but we're most likely talking about a receptor on the surface of the cell. Okay, we'll have acceptance of that, but you've got a receptor here, so think about there being a cell membrane, things bind, the receptor will make something or something will happen here. We'll talk about different ways that happens. Something happens and then we have the effect. So the first characteristic is going to be specificity. So receptors don't just bind to anything and have their effect happen. Okay, so we've got a receptor, it will bind to this shape, S1, but it won't bind to the S2 shape. Okay. Your insulin receptor binds to the shape of insulin, not to the shape of glucagon. And we'll end up with the effect. Okay, our second thing is we have amplification, and this is kind of minimized in that we've got one friend tells, 3 friends tells, each of them tells 3 friends. It's probably more than that. It's probably 10 or 100 or 1,000, right? So I have my receptor binds here. let's say it's a receptor enzyme and it turns on, it's going to kinase, it's going to phosphorylate things. For every insulin that binds, my insulin receptor will maybe phosphorylate 1,000 of the next thing. Okay? We'll find out in a minute what that is. But so by 1,000, it'll activate 1,000 of the next thing inside the cell. So let's go with our NO. NO comes in, it makes it results in 1,000 cyclic GMPs being made. Cyclic GMPs then turn on another enzyme that makes 1,000 of something else. So now we've gone from 1,000 times 1,000 to 1,000,000 of those signal molecules. Does that make sense? Here it's 1 to 3 to 9. But it's probably more than that. But so you can end up with a big cascade of these things, and you often do. You are not, so here for all of these steps, so let's say this is this arrow, but if that's a phosphorylation, right? This is then making a phosphorylating 3 enzymes that each of those phosphorylates 3. Does that make sense? Okay. So you could have something bind to something, we'll see that in a bit, but to have an amplification, you pretty much have to have an enzyme. Okay. Something binding to something else might cause that something else to amplify, might turn on an enzyme, but that's not an amplification step. All right, desensitization or adaptation. So we talked about this in terms of various things. You've got, here we have it with a receptor, but we talked about this with our the Viagra example, right? With the Viagra example, something gets turned on, literally, and then it needs to be turned off, right? You don't make cyclic GMP and just leave the cyclic GMP on around so those blood vessels stay dilated. All right. In terms of a receptor, you see this actually in the acetylcholine receptors in nerve cells. You may have heard of those before, right? Acetylcholine binds to, if you have a junction between 2 nerve cells, so let's say it's like this. I'm going to do horrible nerve junctions. All right, so we've got this gap here that acetylcholine particles end up in. Acetylcholine gets released in there, they're molecules. The acetylcholine binds to its receptor on the other side, but the acetylcholine receptor's got an acetylcholine esterase activity, and it will break down the acetylcholine. So it gets turned on by having acetylcholine bound to it, and then it's also going to turn itself off by breaking down the acetylcholine, if that makes sense. Okay. So you get a nerve signal, it turns on, and then it needs to be turned off. Otherwise, you can think about it, well, it's what goes on in my feet. They're always firing. Okay. Right? You could imagine you put your clothes on and you'd be feeling your clothes all day, right? The signals turn on and then they get desensitized. Okay, integration. This one's maybe a little more complicated, but a cell has multiple receptors on it and can get multiple signals at the same time. One way to think about that is I could have, let's say I have adrenaline. So adrenaline is our fight or flight hormone, right? Plus, let's say I just had a big meal, insulin. Okay? So this is just an example of how we could think about this. We've got a receptor, and one receptor might be signaled on. The other, when we signal this receptor, it might cause a pathway to be upregulated. We signal this receptor, it causes this other pathway to be downregulated, right? They don't have to be opposing each other. You could have 2 that go the same way as well.
I would say, isn't that like in like, I forget, but it's like there was a question in one of the practice exams where it was like the fatty acid, like melancholy would inhibit something because the
Right. So you could have multiple signals. So malonyl-CoA, having excess malonyl-CoA would inhibit fatty acid breakdown. It was probably the carnitine transporter. I don't remember the problem, but malonyl-CoA means you're making fatty acids, so you don't want to break them down.
Yeah.
So it's sort of like that. You could have multiple things going, but this is on the outside of the cell, right? So if I'm using the adrenal insulin, so what's adrenaline going to do in terms of glycogen? Going to make glycogen when you have adrenaline, or you're going to break glycogen down?
Break it down.
You're going to break it down because you need energy to fight or run, okay? But let's say you just had a big meal. What's insulin going to say you should do with glycogen? Build it up, right? And so depending on how much adrenaline you have compared to how much insulin, you're going to end up with the path, but you're either going to break down glycogen or you're going to make glycogen. Does that make sense? So if you can have a little bit of adrenaline, right? I'm assuming that the way that works is adrenaline wins. Okay, but there'll be something like that. So you've got various systems where you've got a cell that will get multiple inputs and then make some decision based on how those inputs sum up. If I asked you a thinking question about this, they would go the same way. If that makes sense. Because you're not a priori going to know how they're weighted. Questions? Okay. So then we have 6 types of signaling here. 6 types of signaling we're going to talk about. And we're really going to emphasize 3 of these. So I'll start up here at the top. So these first 3 are going to be the most important for us in terms of knowing. Okay. Most important for this chapter, most important in terms of how they show up, and you're most likely to see someplace else. So the first one is a gated ion channel, and this is what we talk about in terms of nerve signals. It's used other places as well. But you can imagine that the substrate right here, or the signal molecule, let's call that acetylcholine. Acetylcholine can bind to channels, cause them to open, and cause ions to come through. Okay. Actually, I don't know that acetylcholine does that. The acetylcholine receptor. Anybody know? Is it G-coupled? Okay. I'm not sure if it signals that or it signals something in the cell that causes other ion channels to open. But you can have gated ion channels. So you've got an ion. This can open and it can let through. It can let through ions when the signal molecule is found. Okay. Our second one here is going to be a receptor enzyme. So we've got insulin does this. Insulin is, we've got a substrate, insulin in this case, that binds, and that activates an enzyme in the inside of the cell. In the case of insulin, it's a kinase that's going to phosphorylate some things. We'll go over that pathway in much more detail in a minute. A serpentine receptor. I want to also call these, you'll need to know this name as well. I'm more likely to use G-coupled protein receptor. Whoops. All right, so we got the G-coupled protein receptor. The reason why these are important is I worked in drug development, or I worked in a company that served drug development when I was in industry. This is 3-decade-old information because I haven't really looked at it since. But it used to be that 80% of drugs were targeting G-coupled protein receptors. Okay, so huge in terms of at least where drugs used to be. They may be more specific now, or they may not. I don't know what the percentage is at this point. But what we have here is we're going to have our signal molecule bind, and these are important, or they stand out in that we're going to have this G protein here that's going to be involved in activating an enzyme that's probably making a second messenger. Our example is going to be cyclic AMP if we get to it today. Okay, those are the three most important, and we're going to talk about them in more detail. All right, we can also have, we've talked about how sterols, if I scoot down here, how sterols go right through the membrane. So the steroid hormones, testosterone, estrogen, cortisol, a bunch of these, that they don't have a receptor out here. They go directly through the plasma membrane and the nuclear membrane, and they actually hit, you've got a receptor here. This may seem a little weird. Your receptor is like floating around in solution. The sterol binds to it, it activates it, and then we'll have something happen here. Okay. And we'll see this in our insulin pathway, that not that you get this, that you actually have this floating molecule gets activated. It's not a sterol related. Maybe that's not, insulin is not a good example there. Okay, then we can have receptors that don't have any particular activity. So what happens here is you have your receptor, it binds to a molecule, it changes shape, then our kinase will bind to, let's say a kinase will bind to it and change shape, and then that will make some things. That makes sense. Okay. So I'm going to talk about amplification later today. This step right here, this step to this step, if we think about these as steps, think about that binding and activating is 1, making phosphorylating, this is 2. This is not amplification. 1 receptor binds to 1 enzyme. You're not amplifying. That 1 enzyme then phosphorylates 1,000 and 1,000 other molecules. That's an amplification step. Does that make sense? Okay. People often get confused on that. And then we have adhesion receptors. So 2 of these things, we give 2 things on the cell membrane. When they interact, they stick together based on interactions with the outside world, and that can result in changes in the intracellular matrix, or the extracellular matrix, it says. But it's going to change the cytoskeleton. Questions on any of those? That was sort of general. Okay, let's go on then. And we're going to talk about what's next. Oh, the insulin receptor. Okay, so we're going to talk about the insulin pathway for gene expression here. So if you're well fed, you have insulin around. If you're well fed for a while, that means that you've got enough energy to grow. So I'm going to show you what this whole thing looks like, and then I'm going to make it bigger so we can go through it one at a time. Okay. So this is the insulin pathway for gene expression. There are other insulin pathways. So insulin pathway for gene expression. Okay, so this is used for growth. So this is not a, hey, we're going to store glycogen, right? We'll get insulin, we're going to store glycogen, we're going to break down a glucose. Insulin will signal that in other ways. Insulin is an example of a receptor enzyme. It's the textbook example. Okay. Just like hemoglobin is the textbook example of protein structure and function. So what happens here, we're going to walk through, I'm just going to use their steps and I'll maybe annotate them as I think is useful. Insulin binds to this receptor. You have to have 2 insulins bound, that's not really important, but insulin's going to bind to the receptor, and it's going to cause the kinase portion of insulin to become active, okay? What actually happens is insulin these 2 receptors phosphorylate each other, but we're activating a kinase here. All right. The important step here where we have, so we're activating a kinase, but is that, Is that an amplification step? No, we're turning on one enzyme. So one insulin turns on one enzyme, or two insulins turn on two enzymes, right? So then here we have this thing called IRS, which is insulin receptor substrate 1. Let's name that because it's super important. IRS equals IRS1, insulin substrate receptor 1, not creatively named. Okay, this is what we like to call a protein second messenger. Okay, so like cyclic GMP or cyclic AMP is a second messenger, here we have a protein that gets phosphorylated, and then things sense, other pathways sense that it's phosphorylated, and you have other pathways come off of it. So this is one. We'll talk about the pathway that helps make glycogen is another. Okay, and there are more than that. Is this an amplification step?
No.
Yes.
Yes.
Yes. I have an enzyme that's going to phosphorylate 10, 100, 1,000 insulin receptor 1s, right? One of these can make 100,000 of these, 100 or 1,000 or whatever of these. Okay? Does that make sense? Often there's a lot of confusion on that. So this is, I'm going to do this, amplification steps. So this one is an amplification step. All right, so we have a bunch of pathways that can branch off here, but we're talking about one of those pathways. So we have this protein GRB2 that's going to bind to the insulin receptor substrate if it's phosphorylated, but only if it's phosphorylated. GRB2 is going to change shape because of that. Amplification step or no? No. One IRS2 binds to one GRB2. Okay? We don't activate. You're activating the GRB2 by causing it to change shape, but you're not activating more than one because it's going to stay bound. Then we have this SOS thing that can bind to the GRB2 that's in a different shape. The SOS2 is in a different shape. Okay? And that can be bound by RAS. Okay, this is a bunch of alphabet soup, but we do want to, you're going to have to know this one because we're going to you need to know how a couple of these pathways work so you can think about them in terms of other pathways. So RAS is going to bind, it's a G protein, so it can bind to, when it gets bound, it can hydrolyze GTP. When it gets bound, it can bind GTP. Okay, so GRB2 then to RAS causing the GDP release and bind, and this, so we're going to get rid of the GDP, we're going to get a GTP in here. Now this changes shape, and it can bind to, RAF1 can bind to it and become activated. Okay? Is this an amplification step?
Mm-hmm.
No, because I have one binds to one. So this is not amplification all the way through here. This binds to this. One IRS binds to one GRB2. One SOS binds to one GRB2. One RAS binds to one SOS. One RAF binds to one RAS. And then, how about this one? RAF1 will then, it's activated, it's a kinase, it's going to activate MEK by phosphorylating. So that's an amplification step, right? One can make many. All right, I'm not going to care that you know the full names of these. This is mitogen something kinase. Okay. So mitogen something kinase here is going to activate whatever ERK kinase is. Okay. See, it's also an amplification step. And then ERK can go into the nucleus, and it's a kinase, and it could activate ELK by phosphorylating it. Okay? So also an amplification step. Every time that we're adding phosphates to these things, didn't count for the one above, but up here we've got anytime we have a kinase come in and activate that. And then this will come together with whatever SRF is, and they'll bind to DNA and cause the synthesis of new mRNA for new proteins. Yes.
Do you care if you know the residues?
The residues, meaning the I don't know what you mean by that.
It's saying phosphorylate ERK on the three.
I don't care whether whether it's phosphorylate, a threonine, a tyrosine, a serine. Okay, various things happen there. I do care that so the way I've asked this in the past, I'll give the order and I'll maybe ask for some description of what's going on for some piece of it. Okay, so give me these three, what goes on. And so if I asked you that for SOS through, let's say, RAF1, you say SOS binds to GRB2, changes shape, that's important, right? Changes shape, it's then bound by RAS, that's going to bind to change shape and bind to GTP, which gives RAS the shape that RAF1 is then going to bind to and become activated, and RAF1 will phosphorylate MEK. And then I'd stop. That makes sense. Questions? Okay, so the deal here is this: we started with insulin, we had several amplification steps, and what we end up is in the nucleus we start expressing genes that are involved with growth. Okay. Let's talk about, I've got a G-coupled protein receptor here next. I think let's go ahead and I've changed my mind on how I set this up. Let's talk about, I need to find the pictures for, let's talk about glycogen synthesis. Here it is. All right, so this one's different, and it's got some interesting bits in it. So this is the pathway involved in glycogen synthesis. I think I can fit this on and we'll be able to read everything. Yeah, okay. So this pathway starts also with IRS1 phosphorylated. What happened before this? Don't really need, in general. Do you remember that? So we have, I'm just going to say C above for insulin binding. Right, insulin binds, it causes the autophosphorylation, which activates the kinase part of the insulin receptor. And so the enzyme, it's called, say, enzyme receptor, but that kinase is activated, it's going to phosphorylate IRS1. Okay, and then, okay, what we end up with is this IRS1 that's phosphorylated can be bound by PIK3, which causes PIK3 can then take PIP, so it's PI3 kinase. It can take a lipid. This is kind of interesting because we have a lipid here that's called PIP2. I forgot to put the structures on, but essentially it's got two phosphates on it, and then it gets a third phosphate. And then this lipid, it's involved in all sorts of different types of signaling. We've got a lipid that has the extra phosphate on it. That can be bound by protein kinase B. Okay, once protein kinase B is bound, it changes shape. It's over here such that we don't have PDK1 shown here, but it's going to get phosphorylated and activated. Or actually, let's see. It gets, yes, it gets activated. Okay, and then here's the interesting thing that is different than what we've seen, right? When you phosphorylate something, what do you, in your head, something gets phosphorylated, what happens to it? An enzyme gets phosphorylated, what happens? It gets activated, but not here. Okay, and that's part of why I like to show this pathway. So what we've got here is we've got this enzyme, glycogen synthase kinase, that when it's not phosphorylated, it's active, right? Not phosphorylated, okay? It's active, so it's busy. When it's not phosphorylated, it's busy phosphorylating glycogen synthase to turn it off. So glycogen synthase, we have to back up a couple steps. Glycogen synthase is turned off when it's phosphorylated, and it's being phosphorylated by glycogen synthase kinase. So this pathway up here, what it does is it phosphorylates glycogen synthase kinase to turn it off, to make it stop turning off glycogen synthase, and then glycogen synthase now can make glycogen. Does that make sense? Okay. And there's also an effect to make more GLUT4 transporters to help glucose get in. We're not going to worry about that. Yes? What's the big picture? Yeah. Well, so when insulin binds to the cell, if you've got insulin, you've had a big meal, you have a lot of blood sugar, you should start making glycogen. And this is the pathway, there's another pathway by how that happens. And this is an example of how insulin substrate 1 is a protein second messenger. We phosphorylated it, and I've given you two pathways that get started at that point. And there are more, and we won't go over them. So I realize it's a lot of alphabet soup. I do want you to know these two pathways. In particular, the part of this pathway that I think is really important, right? You don't see a lot of lipid things. That's kind of cool, but that's not as important as the fact that here we have an glycogen synthase being actively turned off by phosphorylation, right? And the way you activate this system is you stop it from being turned off. And by doing that, you have to phosphorylate the enzyme that was turning it off. That makes sense. So I phosphorylate this to turn it off, so I stop phosphorylating this and turning it off. Okay. That's the most important thing I would ask you about. That's probably what I would ask you about for that. Questions? All right, let's talk about G-coupled protein receptors then we'll be done. You won't have to see me in person for a week after that. All right, so this is an example. So here we've got an epinephrine receptor. So this is adrenaline. Epinephrine and adrenaline are the same thing. What happens here is it's one of these serpentine receptors, but that's not really the important part. The important part is what's going on with these G proteins. All right, and so what we're going to have here is something's going to bind here. Epinephrine's going to bind here. The G proteins are going to do their thing, and we'll talk about what their thing is, and that's going to activate adenylyl cyclase, which makes cyclic AMP, which is a second messenger. Right? So we have something binding to the outside of the cell that causes an enzyme in the cell to make cyclic AMP. So what happens here then? So epinephrine binds. What am I always going to say when something binds to one of these receptors? It causes a change in shape. Is that what you said? Yeah. Okay, great. I couldn't hear it. But so it causes it to change shape. All these things, it causes it to change shape, right? That's what's going on. And then something could change shape and become an active enzyme, or it could change shape and something else would bind it. That's really kind of the options we're seeing here. Let's look over here what happens with the G-coupled protein receptors. So we've got a chain, we've got a change in shape. What that causes is it causes this G protein, right, this G piece, to dissociate. It binds to GTP and it's now active. All right. So in this case, it binds to GTP, it's active, and in that shape and being active, it binds to adenylyl cyclase and it activates the adenylyl cyclase. Okay. But you have to turn these things off. So it binds to the GTP, and after some amount of time, it's an enzyme. It will degrade the GTP to make GDP, and now it is inactive. It will stop binding to the adenylyl cyclase, and it will come join this other piece here, which is it's made up of a gamma and beta subunit. But it's going to join that piece and come back and stick back to the receptor. Okay. So we've got two figures here. These two pieces, right? This binds. Shape change occurs. These things sense that shape change. They come apart, right? This one, when it's apart from this piece here, is going to bind to GTP and become active. It binds to adenylyl cyclase, and we get our cyclic AMP made. After some amount of time, this is an enzyme, it's going to hydrolyze that GTP to a GDP, and then it's going to come back and bind. But the G-coupled protein receptor refers to this GTP stuff. Okay. It's involved. It's active. It's active when it's bound to GTP, and then it inactivates itself and comes back and rebinds to its enzyme or its receptor. Questions on that or anything from today? Okay. So what we haven't covered then is we had our three types of signaling that were really important, and we haven't talked about an ion channel yet. Okay, a gated ion channel. All right, well, we'll stop there. You guys have a good week.